reprogram the skin to a state that they resemble the embryonic cells that you would expect isolated from early embryos, which harbor the potential to divide unlimitedly, meaning that you can produce 100, 1,000, 1 million, even 1 billion cells, even more than that, as many as you wanted in a culture dish. And not only that, these cells have the potential to generate all the tissues and cell types in our body. There's a neuron, red blood cells, and muscle, just a few examples. And there's an emerging concept in the pluripotent stem cells, is that we realize in the recent years, there is not only one single type of pluripotent stem cells that we can culture them in a culture dish. The reason is the embryo development is a continuous process. And the pluripotency, the property of cells to turn into any tissue type in our body, is not a singular property in time. It gradually changes and disappears at the later stage of the embryo development. Without boring you with much details, there's two major types of pluripotent stem cells that we isolated. One of them is called naive, the other one is called primed. And the naive, meaning that these cells are isolated from an earlier time point during embryo development. And the primed simply means that they are isolated from later stage of embryo development. They are not the same. They have different properties, they have different potentials. But the take-home message is that naive cells are the cells what we needed, because they are isolated from these blastocyst structures, which I mentioned earlier. Therefore, they have the similar properties of the cells that you would expect from the inner cell mass, which has the potential to give rise to all the tissues and organs. And not only that, if you put these naive iPS cells that generated from your skin and put it back into the early embryos, they can contribute to the embryo development and give rise to a chimera embryos. And with the cells contributing by these naive iPS cells, which labeled in the red color here. So with these two technology at hand, now we can combine them and to generate the tissues or organs using the blastocyst complementation system. So as I mentioned earlier, the genes to change and how to change them, we use one-cell gene editing. And the cells to use, we use naive iPS cells. So I want to give you a few examples by using the mice and the rat, two different species. The reason why we use two different species is that in order to generate the human organs, we cannot use human embryos to do this work. It's ethically forbidden. We have to choose another animal host. To demonstrate efficacy of this approach, we first use the rat and mice, two different kinds of species, whether this can work or not. So for proof of concept, we collect the one-cell embryos from the mouse. We inject them with the gene editing components, which they can modify the gene called PTX1 here again. It's important for the pancreas development. And we grow them to the blastocyst stage. And at this stage, where you have the inner cell mass, the cells here will lacking the gene PTX1, which is going to give rise to the pancreas. And then we use the rat iPS cells, the cells from a different species from the rat. Then we can rescue this blastocyst and generate the rat and mice chimeras, which eventually all the pancreas will be coming from the rat, which labeled with the red color here. Here again, just to show a few more pictures demonstrating that this possibility. If you take out the PTX1 gene, you're lacking a pancreas. This is normal how pancreas looks like in the body. And if we rescue with the red iPS cells, which labeled with red color, you can see all the pancreas is coming from the rat. And this is just a better resolution pictures showing this the pancreas generated by using the red iPS cells labeled with the red color. And this is the markers showing these cells are actually functional. They have the proper expression of an enzyme that normally expressed in the pancreas. And I'm going to give you another example, the eye. So this is the mouse embryo without eye. The reason why it doesn't have an eye is because we take out one gene called PAX6. And if we rescue this mouse with the red iPS cells, as you can see here, it's glowing, is where the red cells are labeled. And they can contribute to the development of a mouse eye. And these mice can be born, and they will have an eye that entirely coming from the rat. So, of course, our goal is not to cure a rat. We want to work on the humans. And I'm just taking this picture drawn by Ernest Haeckel a century ago. And the idea is, during early development, humans and other animals are not so much different, are very similar. Here are just the real pictures. This is one I borrowed from the internet. This is a 6-week-old human embryos. On the left is the pig embryos at 4 weeks old. If I don't label them, probably you cannot tell the difference. They have a liver, they have an eye, they have a spine, they have lymph. So that's why we think the pig may be one of the best models to be used as a host. And not only that, but also the organ size and physiology of a pig is very similar to human. And there's another reason, is you might believe eventually we're all going to go there, sooner or later. You know. So same in the pig, what do we do? There's 2 technologies we need to consider here. Is the genes to change and how to change them. I'm going to first talk about that. So we have a wonderful collaboration at UC Davis and also from Spain. They have a big pig farm. We can collect a lot of zygote, the one-cell embryos from the pig. And we can deliver the gene editing components directly into the pig zygote here. It's very simple. It just basically insert a needle inside those embryos and deliver a small drop of liquid. And this small drop of liquid will contain everything you needed to editing precisely at the location in the genome of the pig. And after that, we choose our host, which is a normal-sized pig, 250 kilo. I have to go there to help carry the pig to the surgical platform shown here. So we have a team of surgeons help us to operate the pig. And here just a video showing that during the operation, this is the final procedure. They make a small opening in the uterus of the pig and then transfer the embryos that we edited inside the pig. Although this is very tiny, it contains about 40 pig embryos inside. So after that, at day 28, we isolate the pig embryos. They are about 2 centimeters in length. And when we analyze it, actually we can delete the gene entirely from the pig, the PDX1. So showing on the left, the green cells are the cells that are expressing PDX1, that are PDX1 positive. And after we deleting, using our gene editing tools in the early embryos, we ended up with a pig that doesn't have this type of cells. And as a consequence, on the top, this is a small pancreas generated inside the day 28 pig embryos. But when we delete the gene, you see this pancreas is disorganized. So with further development, this pancreas will be entirely gone. So the niche will be entirely opened to accept human cells, to be able to generate an entirely human pancreas inside this niche. So after that, we know that we can modify the pig genome just like we did with the mouse very precisely. We can take out any single genes involved in any particular organ or tissue development. What about the cells to use? So bear in mind that we're working with two different species here. And human and pig, the cells, they don't grow at the same pace. They have a different cell cycle. They have a different characteristic. So how do we merge them together? The slides showing here is that the timing to match the human cells to the early embryonic cells of the pig is critical in this experiment. It's just like you're merging into a highway. You need to choose the right time. You need to step on the gas, or you need to step on the brake to find the opening to where you can enter into the highly crowded highway. Simple like that, but it's not so simple. Without giving you too much of the information, I'm just going to say that the naive human iPS cells are the cells that are likely to work. The reason why? So we generate this type of cells. We label with the green color here so we can distinguish the human cells versus other species. First, we test these human cells inside the mouse blastocyst. So we inject these human cells inside the mouse blastocyst, and as you can see, this clump of human cells are locating at exactly the location where we want them to be, which is the inner cell mass. The inner cell mass, as I remind you again, is a cell type that's going to give rise to every single tissues and organs in our body. So, meaning these cells can find the right location and be able to integrate and engraft. Of course, we're not going to use mouse because the mouse is too tiny, the organs are too small. What about the pig? Here I'm going to show you a video, and this is at the time that we inject the human cells into the pig blastocyst. First, we need to use the laser to create the opening of the layers. And then we insert this needle into this cavity here, which is now entirely clear because there is a lot of lipids outside. And these are individual human cells, human naive iPS cells. That we injected about 14 of them in this case. And after that, great. We also find the human cells inside the inner cell mass of the pig as well, where the inner cell mass of the pig can give rise to the whole body, organs, tissues of the whole pig, and with the human cell engrafted. And at day 28, when we isolate the pig embryos, we still see the human cells persist in the pig embryos. And with further analysis, we can see these human cells are intermixed with their endogenous pig cells, meaning that they're at the right location to give the right tissue. So what's next? Now we have the gene editing tools, we have the cell type to use. Now the next, we want to combine them together to see whether we can generate functional pancreas in a pig. I'm sorry to say that we are not there yet. But this is what I imagine the whole system going to work. So we collect the pig embryos, particularly the one-cell embryos, the zygote, the fertilized egg, and we deliver the gene editing components, which you saw in the video, a small drop of liquid contained everything we needed to delete a gene that we wanted in the pig. And then we culture them to this ball of cells called blastocyst again. And we take a skin from you, and we turn them back into naive human iPS cells, pluripotent stem cells. And we inject the human pluripotent stem cells, which generated from your skin, into the blastocyst of the pig. And then we choose the host of the pig and perform embryo transfer. Then after the piglets are born, we can harness the organs and tissue, the cells that originally coming from your skin. So in case you need it in the future, we don't have to go to the black market again. We can directly use these cells that generated from your skin and to cure any disease that you may have in the future. So with that, I'd like to thank all the people that are involved in this project. And these are the group of people working in our lab. And these are the collaborators from UC Davis, and also the surgical team, and also the hospital from Spain. And there's 2 pig farms that help us a lot for this type of experiment. And most importantly, I'd like to dedicate this work to Erwan Zhang, who sponsored our research. Without his support, there will be no progress as fast as I presented today. So I'd like to end with one sentence that's coming from Erwan. There are so many creative people that if you just give opportunity and little guidance, you will change the world. Thank you.
Thank you, June. Is this mic working? Yes, great. Well then, I will be happy to pass this to anybody in the audience if you have questions, because I know that my mind is exploding with the future of medicine and lots of questions. Any takers? Okay. All the way.
Hi. Do you anticipate making organs on demand to match a particular individual's genome, or is there a way to, during your pluripotent stem cell stage, of maybe reducing the antibodies so that they're more generally accepted by anybody who needs the organ? Or
That's a
would
great
you have
question.
to go through the normal immunosuppression that we do now with transplants?
I think we are approaching in both directions. So the most ideal situation is we want to take the cells from you and generate the organ that come from your genome, which can minimize the immune response if you would expect them to transplant back to you. But also we understand that this procedure may take time. We can choose the alternative way, which we use the pluripotent cells that are matching to your immune system, but generated in the bank, in the cell bank, and use that for transplantation in the future to minimize the immune rejections. But we're taking both directions.
Okay.
Hi.
Hi.
So when you took a mouse and you grew a rat eye or a rat spleen in it, did you transplant the rat eye or the rat spleen back into a rat to see if it would accept it?
We haven't transplanted the eye yet, but we're in the process of transplanting the pancreas back to the rat. Yes. And we do expect there's a certain degree of immune rejection because we generate the rat cells in the mouse, and the environment is quite different from the rat. But our idea is to combine this with immune suppressant. So in that way, at least the cells we know is matching the cells we're transplanting into. But there are some other molecules that the cells carry. We can suppress that further with the immunosuppressant.
Thank you very much. That was fantastic. In terms of Parkinson's, in terms of your research, do you see any progress at all in that area?
Neurons. So that's a very, very great question. I think for these purposes, we're a little bit farther away from the neuron because now the people are concerning is whether the human cells will contribute to the neural development, which they worried. But we are taking every precautions that we have to prevent these human cells, to guide them only to the particular organs that you wanted. So we are, actually in our case, we are avoiding the neurons. But back to your question, I think there is a lot of effort that try to generate neurons from the human proponent cells in the culture dish. I think there is a lot of development, advancement in their areas. But as I mentioned earlier, if we grow everything in the culture dish, that takes time, and the cell characteristic may not be the same, although they are very similar. But I think with this approach, if it's been proven to work with the pancreas, to work with the liver, to work with the heart, other major organs, in the future, we can consider to generate particular cell type in the neuron systems that can be used for transplantation in such as Parkinson's disease. But we're, at this stage, we don't want to generate the whole brain. We're probably only targeting a particular cell type that we need it for. So.
Is there a size difference if you transplant the rat pancreas into the mouse? Would it be too large, or would it adjust?
Great question. Yes, that's why we choose pig, at least for the human. But with the rat and mice, the size is a big problem. But in the case of the pancreas, we don't need to transplant the whole organ, actually. We can isolate the islet, and we can transplant only the islet to control the insulin levels of the mouse. So we don't need to worry about the size. But with regard to human, I think the size is very important. That's why we choose the pig, which have very similar size, especially the pancreas and the liver and the heart, to match each other.
Okay, one last question before our reception.
Great talk. So I have a question about when you do these procedures and you create the chimera and you're putting like the human into the pig. There are what I believe is epigenetic issues with the development in another species. Does that, or do you know, how will that affect when you put it back in? Will it work? Because now it's been developed in a pig. Is it going to work in the human? Is it going to live, because our lifespan is much longer than a pig. Do you know anything about how the cell is really going to function when you put it back, raising it in a different environment, genetic environment?
That's a fantastic question. That's the question I had 1 year ago. I was worried about the same thing. Because it's, to me, just like you mentioned, merging two species together is just crazy 1 year ago. But throughout these years, we get more information, we know more. And we realize that the nature, the animal development itself, they already figured it out. They will kick all these unfit cells. The cells are not be able to properly contribute to the particular organs or tissues. They already eliminate them. They only keep the good ones. And for reasons we don't really fully understand. I guess that's the same question that I raised in the earlier. You grow the cells in the culture dish, which is in a totally foreign, alien environment than the animal body. And even with that, we can generate the cells that are similar to some extent to the cells isolated from our body. So for that, I think there is a lot of unknown, a lot of things that we don't understand. But the beauty of this system is that we don't have to understand everything before we can generate something useful. And you mentioned perfectly, we don't really know, but we know it worked. That's all important here.
Well, I think that's a great note to end on for today. Thank you again for coming.